Hello students, welcome to the session. In this session, I would like to discuss with you the assignment key. So let us see, in the previous session what we have learnt. We have learnt about the beginning stage of mitosis and some introduction steps which are related to interphase we have learnt in the previous session. Students, in this session, I will discuss with you the major important key as well as solution for each and every question. Let us look at first question. First question is, what is a prophase in meiotic cell division? What is a prophase? As we know that the mitotic cell division contains some of the subphases like mitosis or mitotic, both are same. It contains some of the subphases like prophase, metaphase, anaphase, telophase. Now let us see the first stage called prophase. Here, what is a prophase in meiotic or mitotic cell division? So let us see here. You know that during meiotic cell division, how the cell looks like. How the cell looks like. The cell generally looks like this. So here this is called as an anterior phase or anterior pole. Anterior pole, whereas the opposite one is called as the posterior one or inferior side. Posterior one or inferior side. Got it, Nana? Now let us see during a prophase what are the sequential steps takes place. During prophase, the prophase generally followed by S phase and G2 phase. We know very well about S phase. In the previous session, we have learned about S phase. We know very well about the G2 phase. Already we have discussed in the previous assignment. S phase, synthesis phase, G2 phase, gap 2 phase. About these two stages we have learned already. So, these two stages after we are going to learn the meiotic cell division. In this case, condensation of chromosomes, it's a common character in the meiotic prophase. Suppose take a nucleus, as you know very well, here the nucleus is present or nucleus is seen. The nucleus generally looks like this. Here the nucleus contains genetic material. The nucleus contains genetic material. So the genetic material in front of electronic microscope generally visible like this. So this is nucleus, the nucleus, inside the nucleus, genetic material or some solid substance is present inside the nucleus called genetic material. So this solid substance generally undergoes for condensation process. It undergoes for condensation. What do you mean by condensation? How can you define condensation? During condensation process, the nucleoplasm or nucleus fluid undergoes for some sequential changes. Finally, nucleoplasm disappears and the genetic material completely turns into thread-like fibers or fiber-like structures which are called as chromosomes. So here the condensation process which leads to formation of thread-like fibers or fiber-like structures called as chromosomes. But it is called chromosomes. So here along with these chromosomes, what are other steps or other changes you can see in prophase? You know that centromeres, the centromeres that are present in the cell, these centromeres, you can see the centromeres here, centromeres which you can see in the prophase, these centromeres, they forms a fiber-like structures called centromere fibers. Centromere fibers. These centromeres or centromere fibers, later on they forms spindle fibers, as we know very well about the spindle fibers. These spindle fibers helps in the pulling of chromosomes towards their opposite end. So finally what will happen here at the end of this prophase, because of condensation, genetic material undergoes for condensation, finally produces thread-like fibers called as chromosomes. What are they called? Chromosomes. What is a chromosome? That we are going to discuss in the further step. Students, I am moving on to next question. Look at the next question. Let us see the next question. Second question is, what are the different events in prophase of mitosis? Just now we have learnt. Now let us see what are the different events takes place in prophase. First event is initiation of chromosomes. Initiation of condensation of chromosomes. So when there is a condensation process, finally it leads to formation of chromosomes. That is the first stage, first event we have seen in the prophase. So how these chromosomes are formed? These chromosomes are formed from a solid substance that is seen in the nucleus called genetic material. So what I am trying to say, condensation process takes place in a separate solid substance called genetic material. When this genetic material undergoes for condensation, it leads to formation of fiber-like structures called chromosomes. Next stage or next event, what occurs in the prophase? Next step, movement of centrioles. As I said to you, the centrioles, if you take the centrioles, the movement of centrioles you can see. The movement of centrioles, where they moves, to which place they move. The centrioles generally moves towards their opposite ends or opposite poles. So you know that I have said to you, the cell contains an anterior portion or anterior pole as well as the posterior pole. So these centrioles slowly moves towards their opposite end. These are the sequential steps that takes place in the prophase. One more event also we can see in this prophase. What is that one more event? Disappearance of genetic material, which means in the cell or inside the nucleus, whatever the genetic material is there, this genetic material transforms into chromosomes. That is also one of the steps, isn't it? That is also one of the event. Along with this, the cell that contains some special cell organelles like endoplasmic reticulum that already we discussed in the previous sessions, and also Golgi apparatus. These two cell organelles completely disappears during prophase. Along with this, one more event also occurs, that is disappearance of nuclear envelope. So the nucleus contains a membranous structure called nuclear envelope that disappears at the end of the prophase. Students, these are the sequential events takes place in the prophase. I am moving on to next question. Let us see the next question. What is the next question? Third question. In metaphase, chromosomes are arranged at the center of the cell is called. Let us read the question once again. In metaphase, chromosomes are arranged at the center of the cell is called prophase or prophase plate, metaphase plate, interphase plate, all of this. What is your suitable answer? It's definitely a metaphase plate. Why? Because it takes place during metaphase, hence it is called as a metaphase plate. How this metaphase plate looks like? Suppose imagine it is a cell. At the center of the cell, there is a plate-like structure appears. In this plate-like structure, you can see some special structures called as chromosomes. So, this plate-like structure first appears at metaphase, hence it is called as a metaphase plate. What it is called? Metaphase plate. Clear, children? Moving on to next question. Let us see the fourth question. What is fourth question? In dash phase, it's a suitable to study chromosomes morphology. During prophase, because of condensation of genetic material, chromosomes are appeared or chromosomes are formed. Isn't it? While the same cell enters into metaphase, what will happen? Clear structure of chromosomes appears, which means chromosomes are clearly visible under microscope in the second stage of meiotic cell division. So, what is the second stage of meiotic cell division? It is definitely the metaphase, isn't it? So, the correct suitable option for this is metaphase. Moving on to next question. Let us see the next question. Let us see the next question, students. Fifth question. What is meant by sister chromatids? What do you mean by sister? What do you mean by sisters? Twin sisters. Monozygotic, dizygotic, twin sisters. Here, the question given like this. Sister chromatids. What do you mean by it? Sister chromatids are replicate chromosomes of eukaryotic chromosomes. Suppose take an eukaryotic chromosomes. The eukaryotic chromosomes are generally looks like this. The eukaryotic chromosomes are generally looks like this. So here, these kind of chromosomes, the paired chromosomes are called, or the paired structure of chromosomes are called generally sister chromatids. What are they called? Sister chromatids. Isn't it? So the paired structures, paired organisms, nothing but twins, isn't it? In the same way, sister, they have equivalent characters or identical characters, isn't it? Chromosomes which have identical characters generally considered as sister chromatids. These sister chromatids are replicate chromosomes of eukaryotic organisms. Suppose take an eukaryotic organism. Best example for eukaryotic organisms such as like animals. If you take a human body is the best example for cells such as like eukaryotic cells. In these eukaryotic cells, you can see the sister chromatids that have equivalent genetic material. Hence, they are called sister chromatids. Moving on to next question, students. Let us see the sixth question. Splitting of chromosomes at the centromere into two sister chromatids takes place at which stage of the or which phase of the meiotic cell division? So, just now I have shown the paired chromosomes or doubled chromosomes. So, they will be later on splitting into equal chromosomes at which phase of the meiotic cell division? Let us see. So, during second stage of meiotic cell division, we have seen the paired or sister chromatids, isn't it? What is the third stage? So, the third stage is anaphase. During anaphase, the sister chromatids, suppose take these are sister chromatids, the sister chromosomes or paired chromosomes. The sister chromatids, they will be separated into equal chromosomes in a third phase of this meiotic cell division called anaphase. In which phase, Nana? During anaphase. So, how this anaphase stage looks like? Look at the cell. Imagine it is a cell. So, from anterior portion of the cell and posterior portion of the cell, fiber-like structures appears. What are these fiber-like structures called? Spindle fibers. What are they called? Spindle fibers. Now here, with the help of these spindle fibers, the sister chromatids are separated into individual chromosomes. Now, the center of the chromosome attaches to this spindle fiber. The center of the chromosome stick to this spindle fiber. So, like this, the chromosomes will be sticked or attaches to this spindle fibers. So after sticking to the center of the chromosomes, the spindle fibers pulls these chromosomes towards their opposite end, isn't it? That we know very well, already we have seen. So anyway, these are the sequential stages or steps or events that takes place in Anaphase. So Anaphase is the suitable answer for this question. Moving on to next question, students. Seventh question. Complete disappearance of nuclear membrane takes place in dash phase. In which of the following phases complete nuclear envelope disappears? I already discussed with you in the previous question or in the second question or third question. The nuclear membrane disappears in metaphase. In which phase? Completely it disappears in metaphase. What is the correct answer? So the correct answer is the second option, metaphase. Isn't it? Direct question. Moving on to next question, students. Eighth one. What is anaphase? How can you determine or how can you explain the process of anaphase? Let us see the anaphase. So anaphase is the third stage of the cell division or meiotic cell division, in which some of the sequential changes occurs. If you look at the anaphase, from anterior portion of the cell and posterior portion of the cell, fiber-like structures appears. These fiber-like structures are called as a spindle fibers. These spindle fibers stick to the center of the each chromosome. So when they stick to the center of the chromosomes, what will happen, you know? These spindle fibers pulls these chromosomes towards their opposite end. Okay? So these are the general sequential changes that occurs in anaphase. At the end of the anaphase, you can see the small projection that appears at the center of the cell. Okay? So what is the importance of this small projection? So we are going to discuss in the telophase. So anyway, these are the important questions, students, which are given in our assignment key. I hope so everybody understood this assignment key, exciting assignment key. Remaining assignment keys we are going to discuss in the further classes, further sessions. Thank you. Be safe. Have a nice day.